All right, let's get right into it. Today, we're diving deep into what is essentially the command and control center of your immune system. We're talking about T helper cells. These are the real masterminds, the ones that dictate the entire strategy for your body's defense force.
So just think about it for a second. When a virus or some nasty bacteria gets in, how does your body actually know what to do, right? Who gives the order to attack? And maybe even more importantly, what kind of attack should it be? This is the fundamental challenge your immune system has to solve literally every single day. And the answer? Well, it lies with a very, very special type of cell.
So let's meet the generals of your adaptive immune system, the CD4 T helper cells. Now what's interesting is they don't actually fight on the front lines themselves. Nope. Instead, they act like commanders. They receive intelligence, they assess the threat, and then they coordinate this highly specific response, telling all the other immune cells exactly how to engage the enemy.
To really get what they do, let's look at the whole team. First, you've got the cytotoxic T cells, the CD8+ cells. These are your soldiers, the ones doing the direct hand-to-hand combat. Then you have the regulatory T cells, or Tregs. Think of them as the diplomats. Their job is to calm everything down and prevent friendly fire. But for today, our focus is squarely on that central figure, the CD4+ helper T cell, the general who's calling all the shots.
Okay, so a general isn't just going to launch a full-scale war based on some flimsy rumor, right? They need confirmed intelligence, crystal clear orders. Well, for a brand new, naive T cell, this process is called the three-signal hypothesis. And it's an absolutely critical safety measure that ensures the immune system doesn't accidentally attack the wrong target, like, yourself.
And here's the most important takeaway. Something you have to remember. All three signals are absolutely required for this T cell to get activated. It's non-negotiable. Think of it like this. Signal 1 is recognition. The T cell sees a piece of the enemy and asks, "What is this?" Signal 2 is confirmation. It's like a second handshake that says, "Yes, this is a real threat, you have the go-ahead." And finally, signal 3 is specialization. This is the specific battle plan delivered by molecules called cytokines that tell the T cell exactly what kind of general it needs to become. Now this is where we get into the nitty-gritty, the exact molecules involved. This is prime exam material right here. Let's walk through it. For signal 1, you see the antigen-presenting cell, or APC, uses its MHC class II molecule to show the antigen to the T cell's receptor, the TCR. For signal 2, that confirmation step, the APC CD80 or 86 molecule has to connect with the T cell CD28. And for signal 3, the APC just releases these cytokines that then bind to the T cell cytokine receptors. It's a super secure three-step molecular handshake. So once that third and final signal is received, that T helper cell is ready to choose a career path. It differentiates, it specializes, and it becomes the type of general that is perfectly suited for the specific threat that was just identified. And remember, this entire decision is dictated by the mix of cytokines it was exposed to.
This slide right here, this is your ultimate cheat sheet for the T helper subsets. It's packed with high-yield info. Just look at the inducing cytokines column. These are the molecules that determine the cell's fate. For instance, if the cell sees IL-12, it's pushed to become a Th1 cell. But if it sees IL-4, it's going to become a Th2 cell. Then, these cytokines flip on a key transcription factor. You can think of that as a master genetic switch, which permanently locks that cell into its new identity and job.
Now let's zoom in a bit on the two classic, and you could argue most important subsets, Th1 and Th2. Because as we're about to see, they exist in this really delicate balance, and they're constantly competing with each other for control of the immune response.
And this right here is the heart of their rivalry. It is literally a cytokine tug of war. The main cytokine that Th1 cells make, interferon gamma, actively blocks Th2 cells from developing. But it's a two-way street. The main Th2 cytokine, interleukin-4, does the exact same thing back to the Th1 cells. So as soon as one side gets a little bit of an advantage, it actively suppresses the other one so it can dominate the entire response.
This whole competition is basically hardwired into their genetics. Check it out. On the left, interferon gamma turns on that master switch we talked about, a transcription factor called T-bet. T-bet locks in the Th1 identity. Meanwhile, on the right, interleukin-4 turns on GATA3, which is the master switch for the Th2 identity. Each switch not only turns its own pathway on, but it also helps turn the other pathway off.
So why does this balance matter so much? Well, because they have completely different jobs.
Th1 cells are the experts in cell-mediated immunity. They basically supercharge macrophages to go hunt down and kill pathogens that are hiding inside our own cells. Th2 cells, on the other hand, are in charge of humoral immunity. They're the ones that help B cells churn out massive amounts of antibodies, which is great for fighting things like parasites, but they're also the main drivers behind allergic reactions like asthma.
It really is like the yin and yang of the immune system. You absolutely need both, but they have to be in balance. If you have way too much Th1 activity, you can get autoimmunity. But if you have too much Th2 activity, you can get severe allergies. A healthy immune system is all about picking the right response for the right enemy at the right time.
Okay, let's quickly touch on the other players. First up, 17 cells. These guys are the rapid response inflammation specialists. They are fantastic at fighting off things like extracellular bacteria and fungi because they're great at recruiting other immune cells to the scene of the crime. But because they're so powerfully pro-inflammatory, they're often implicated when the system goes haywire in autoimmune diseases like rheumatoid arthritis or psoriasis.
And finally, we get back to our diplomats, the regulatory T cells, or Tregs. Their entire job is to keep the peace. You can identify them by their master factor, FOXP3. And what they do is shut down immune responses once the threat is gone. They release these inhibitory cytokines like TGF-beta and IL-10 to prevent our immune system from attacking our own body. They are absolutely critical for maintaining what we call self-tolerance.
So we have this elegant, this highly specific system of generals and special forces. It's beautiful. But what happens when an enemy finds a way to just bypass all of that control? What happens when it triggers a system-wide override? Well, that's when we face the superantigen threat.
The whole danger of a superantigen is that it's non-specific. See, a normal antigen is like a very specific key that only fits one lock, activating only the T-cells that are meant for that one threat. But a superantigen, it's like a skeleton key. It just jams the whole system, forcing a connection between the APC and a huge number of T-cells that have nothing to do with the actual infection.
This chart just shows you the terrifying scale of this problem. A normal infection, it might activate something tiny, like 0.01% of your T cells, a very targeted, special ops kind of mission. A superantigen, though, can activate 20, even 30% of all your T cells, all at once. It's a recipe for utter and complete chaos.
And this isn't just some textbook theory. We see these in the real world. Exogenous superantigens, meaning they come from the outside, include those infamous toxins from staph bacteria that cause toxic shock syndrome. And there are also endogenous ones, made inside our own bodies, by viruses like Epstein-Barr, which actually uses this trick to manipulate our immune response.
So just imagine it. You have more than 20% of your T-cell generals all screaming, "Attack!" at the exact same time. What happens? They release a tidal wave of these signaling molecules. And this event, this overwhelming systemic flood of inflammation, that's what we call a cytokine storm.
The consequences of a cytokine storm are incredibly dire. It can lead to systemic inflammation, multiple organ failure, and tragically, it can lead to death. The very system that was designed with such precision to protect us is turned into this indiscriminate weapon against the body itself.
So, we're left with this really profound question. We've seen that the immune system is an absolute masterclass in balance and controlled power. But things like superantigens reveal this critical vulnerability, how that beautiful precision can be hijacked and turned into a fatal flaw. It really makes you wonder, as we develop new medicines to tweak the immune system, how can we learn to harness all of this incredible power without ever unleashing its potential for chaos?